Hello and welcome to Sam Biology class. Friends, today we shall discuss about biological nitrogen fixation. Before that, I request all of my friends, those who have not subscribed my YouTube channel, please subscribe. Biological nitrogen fixation is the reduction of atmospheric nitrogen into ammonia by certain microorganisms such as bacteria or cyanobacteria. It is an essential reaction in the global nitrogen cycle and accounts for 2/3 of the fixed nitrogen produced on the earth. Atmospheric nitrogen can be converted into ammonia by free-living microorganisms as well as by microorganisms which live as symbionts. So there are two types of biological nitrogen fixation. One is non-symbiotic nitrogen fixation and the other one is symbiotic nitrogen fixation. Reduction of atmospheric nitrogen into ammonia by non-symbiotic or free-living microorganisms is called non-symbiotic nitrogen fixation. Example of free-living bacteria which fix atmospheric nitrogen are Bergerichia, Azotobacter, Rhodospirillum, Clostridium and blue-green algae or cyanobacteria such as Nostoc, Anabina, Oscillatoria, etc. The second type is symbiotic nitrogen fixation, which is the reduction of atmospheric nitrogen by symbiotic microorganisms. Examples are Rhizobium, Bradyrhizobium and Azorhizobium. These microorganisms establish symbiotic relationship with a suitable host and fix atmospheric nitrogen. Most of the nitrogen-fixing bacteria are free-living. Several others form symbiotic associationship with higher plants in which the nitrogen-fixing prokaryotes provide fixed nitrogen directly to the host in exchange for nutrients. Such symbiosis occurs within specialized structures called nodules, which contain nitrogen-fixing bacteria. The most common type of symbiosis occurs between the members of the plant family Fabaceae, also called Leguminosae, and soil bacteria of the genus Mesorhizobium, Azorhizobium, Bradyrhizobium, Cyanorhizobium and Rhizobium, which are collectively called Rhizobia. Another type of symbiosis occurs between several woody plant species and soil bacteria of the genus Frankia. Still other types of nitrogen-fixing symbiosis involves South American herb Gunnera and tiny waterfall Azolla, which form associations with cyanobacteria, Nostoc, which form associations with cyanobacteria, Nostoc and Anabina. Finally, some nitrogen-fixing prokaryotes are also associated with C4 grasses such as sugarcane and Miscanthus Symbiotic nitrogen fixation is the part of a mutualistic relationship in which plants provide a niche and fixed carbon to the nitrogen fixers in exchange for fixed nitrogen. Symbiotic nitrogen-fixing bacteria invade the roots of plants and induce the formation of specialized structures called nodules, which contain nitrogen-fixing bacteria. Nitrogenase is the enzyme which fixes atmospheric nitrogen. It contains specialized sites that facilitates the high energy exchange of electrons. Oxygen, being a strong electron acceptor, can damage these sites and irreversibly inactivate nitrogenase. Hence, nitrogen fixation must occur under anaerobic conditions. In cyanobacteria, anaerobic conditions are created within specialized thick wall cells called heterocysts. Heterocysts lack photosystem II, because of which they are not able to generate molecular oxygen during photosynthesis. In cyanobacteria, heterocysts are differentiated when they are deprived of ammonium. By contrast, free-living nitrogen fixers do not establish symbiotic relationship with others, so they live independent of other organisms. Examples of free-living nitrogen fixers are Azotobacter, Azospirillum, Clostridium, Klebsiella and others. These are all free-living nitrogen fixers. Free-living bacteria that are capable of nitrogen fixation are divided into three groups. First one is aerobic nitrogen fixing bacteria such as Azotobacter are thought to maintain a low oxygen concentration through their high respiration. Second one is facultative organisms. These bacteria, which grow under both aerobic and anaerobic conditions, can fix nitrogen only when the condition is anaerobic. Third one is obligate anaerobic nitrogen fixing bacteria, which grow only under anaerobic conditions. These organisms can be photosynthetic or non-photosynthetic. Symbiotic nitrogen fixation occurs within specialized structures. Some symbiotic nitrogen fixing bacteria dwell within nodules, which are specialized structures that develop on roots of plants when infected by nitrogen fixing bacteria. In Gunnera, nitrogen fixation occurs within pre-existing stem glands, which develop independently of the symbionts. In case of legumes and actinorhizal plants, the nitrogen fixing bacteria induce the development of root nodules. Grasses also form symbiotic association with nitrogen fixing prokaryotes, but in such cases root nodules are not formed. Legumes and actinorhizal plants regulate gas permeability in their nodules, maintaining a oxygen level of 20 to 40 nanomolars. This oxygen level can support respiration but is sufficiently low to avoid inactivation of enzyme nitrogenase. Nodules contain oxygen binding heme proteins called leg hemoglobins. Leg hemoglobins are the most abundant proteins in the nodules, crucial for nitrogen fixation. It has a high affinity for oxygen. Its function is to increase the rate of transport of oxygen to the respiring symbiotic bacterial cells, which decrease the steady state level of oxygen in infected cells. To continue aerobic respiration under such conditions, the bacteroid uses a specialized electron transport chain in which the terminal oxidase has an affinity for oxygen which is even greater than that of leg hemoglobin. The symbiosis between legumes and rhizobia is not obligatory. Legume seedlings germinate without any association with rhizobia and may also remain unassociated throughout their life cycle. Rhizobia also occur as free-living organisms in the soil. Under nitrogen-limited conditions, however, the symbionts seek other through an elaborate exchange of signals. Signaling, the subsequent infection process and the development of nitrogen-fixing nodules involve specific genes both in the host and the symbiont. Plant genes specific to nodules are called noduling genes. Rhizobial genes that participate in nodule formation are known as nodulation genes or node genes. Node genes can be classified into common node genes or host-specific node genes. Common node genes such as node A, node B, and node C are present in all rhizobial strains. Host-specific node genes such as node P, node Q, and node H or node E, node F, and node L differ among rhizobial species and determine host ranks. Only one node genes, which is the regulatory node D, is constitutively expressed. The first stage in symbiosis between legumes and rhizobia is the migration of nitrogen-fixing bacteria towards plant roots. This migration is a chemotactic process involving chemical attractants such as isoflavonoids and betaines that are secreted by plant roots. Node factors produced by nitrogen-fixing bacteria serve as signals for symbiosis. Node genes which are activated by node D code for nodulation proteins, most of which are involved in the synthesis of node factors. Node factors are lipochitin, oligosaccharide signal molecules. Three common node genes such as node A, node B, and node C encode three different enzymes with specific functions. Node A encode an N-acyl transferase. Which catalyzes the addition of a fatty acyl chain. Second node gene, that is node B, encode a chitin oligosaccharide deacetylase, chitin oligosaccharide deacetylase that remove the acetyl group from the terminal non-reducing sugar. And third gene, that is node C, encode chitin oligosaccharide synthase, chitin oligosaccharide synthase that links N-acetyl D-glucosamine monomers. The host-specific node genes that differ among rhizobial species are involved in the modification of the fatty side chain or in the addition of groups that are important in determining host specificity. A particular legume host responds to a specific node factor. Legume receptors for node factors are protein kinases with extracellular sugar binding lysine motif domains. These domains are activated by node factors inducing oscillations in the concentration of free calcium ions in the nuclear regions of root epidermal cells. Recognition of calcium ion oscillation requires a calcium calmodulin dependent protein kinase which is associated with a protein known as cyclops of unknown function. Once the calcium ion oscillation is recognized by the epidermal cells, node factor responsive transcriptional regulators directly associate with node factor inducible genes. This results in gene expression changes. Formation of root nodules. Two processes. One is infection and the other one is nodule organogenesis occur simultaneously during the development of root nodules. Rhizobia usually infect root hairs by fast releasing node factors which induce a pronounced curling of root hair. Rhizobia become enclosed in a small compartment formed by curling. Also, in response to node factors, the cell wall of the root hair in this region is degraded, allowing the bacterial cells a direct access to the outer surface of the plasma membrane of the root hair cells. The next step is the formation of infection thread. The infection thread grows at its tip by the fusion of Golgi-derived membrane vesicle at the end of the tube. Deep within the cortex near the xylem, the cortical cells dedifferentiate and start dividing forming a distinct area within the cortex called nodule primordium, which gives rise to root nodules. Different compounds acting positively or negatively control the development of nodule primordium. Node factors activate localized cytokinin signaling in the cortex and pericycle of the root, leading to the localized suppression of polar oxygen transport, which in turn induce nodule morphogenesis and stimulate cell division. Ethylene is synthesized in the pericycle and diffused to the cortex, where it blocks cell division opposite the phloem poles of the root. The infection thread filled with proliferating rhizobia elongates through the root hair and cortical cell layer towards the nodule primordium. When the thread reaches the nodule primordium, its tip fuses with the cell membrane of host cell and penetrates into its cytoplasm. Bacterial cells are subsequently released into the cytoplasm surrounded by cell membrane of host cell. This results in the formation of an organelle called symbiosome. Branching of the infection thread within the nodule enable bacteria to infect many cells. At first, bacteria within symbiosome continue to divide and the symbiosome membrane increase in surface area to accommodate the growth by fusion with smaller vesicles. Soon thereafter, because of an undetermined signal from plant, these bacteria stop dividing and differentiate into nitrogen-fixing bacterioids. Nodules as a whole also develop vascular system for exchange of nitrogen fixed by rhizobia for the nutrients produced by plants. It also develops a layer of cell to exclude molecular oxygen from the root nodule interior. Biological nitrogen fixation is catalyzed by the enzyme nitrogenase complex. Reduction of molecular nitrogen into ammonia, a 6-electron transfer, is coupled to the reduction of 2 protons to evolve S2. The overall reaction catalyzed by the enzyme nitrogenase complex is N2 plus 8 electron and 8 protons plus 16 ATP gives rise to 2NH3 plus H2 and 16 ADP plus 16 inorganic phosphate. The enzyme nitrogenase complex can be separated into 2 components. One component is the Fe protein and the other component is MOFE and the other component is MOFE protein. MO stands for molybdenum and Fe stands for iron. Fe protein is the smaller component. It is composed of 2 identical subunits. Each subunit has a mass that varies from 30 to 72 kilodalton depending upon the bacterial species. Each subunit contains a iron sulfur cluster which participate in the redox reaction that convert nitrogen into ammonia. The Fe protein is irreversibly inactivated by molecular oxygen. Second component is the MOFE protein, which is composed of 4 subunits, and the total molecular mass varies from 180 to 235 kilodalton depending upon the bacterial species. Each subunit contains 2 molybdenum iron sulfur cluster. Molybdenum iron sulfur clusters. Like the other component, this component is also inactivated by molecular oxygen. In the overall nitrogen reduction reaction, pyridoxine serves as electron donor to Fe protein, which in turn hydrolyzes ATP and reduces MOFE protein. Binding of ATP to Fe protein and its hydrolysis to ADP and inorganic phosphate is thought to cause a conformational change in the Fe protein which facilitates the redox reaction. So Fe protein reduces MOFE protein, which then can reduce numerous substrates. But under natural condition, MOFE protein reacts only with N2 and H+, as a result of which molecular nitrogen is reduced to ammonia. So friends, this is all about different aspects of biological nitrogen fixation. For more updates, subscribe my YouTube channel, Sam Biology Class.